Let's look about the VIA, the visual inspection with the acetic acid. Principally, it detects the abnormal cervical epithelium by applying 3 or 5% acetic acid, that is nothing but the vinegar. So what is the procedure? Apply acetic acid to the cervix for 1 minute and observe. What will you observe? If there are white patches, or it is also called as aceto-white areas, which indicates the precancerous changes. So why is this happening? Because in cancer cells there is less amount of glycogen and more amount of protein. Why? Because the cancer cell, they are nothing but rapidly proliferating cells. Their genetic material will be huge. Okay, so they need enough ATP, more amount of ATP. So they will consume all the glycogen stores and it will be depleted. But the protein will be increased, and these acetic acid coagulates with the protein and form the white patches. So after adding the acetic acid, if you see white patches, it shows if there are certain cells which are cancerous or not. Okay. What are the findings? Negative VIA if there is no aceto-white lesions, that is normal. And what if it is positive? Then there will be distinct aceto-white lesions. Possible it is precancerous. What is the advantage of VIA, visual inspection with acetic acid? It is quick, it is low cost and does not require a lab. But the pap smear, this was the gold standard, but it is of a little more cost compared to VIA. Okay. It is used in the low resource setting or an alternative to pap smear in village settings and all. So now you see the pictures. See, you can see the cervix and you can see the white patches. So these are nothing but the acetic positive cervix, which shows probable cancer cells. Okay. At the end, we'll be talking about VILI, that is visual inspection with the Lugol's iodine. Just how we used the acetic acid here, we will use Lugol's iodine here. So what is the principle? Identifies abnormal cervical cells by staining with the Lugol's iodine. So what is the procedure? Apply Lugol's iodine to the cervix. So after you apply, now let's compare. So this part is where the acetic acid was applied, and here you can see the white patches, and in the same cervix you can see a Lugol's iodine was used, and here you will get the yellow patches, right? So normal squamous, if there is normal squamous epithelium, it will be dark. So see, all this part is the dark because those are the normal. Why it is dark? Because these normal cells has good amount of glycogen, because these are normal cells, they will store glycogen. But what about the cancer cells? So cancer cells does not store glycogen. Why? Because they are ATP hungry. They need ATP because they are ATP hungry. They need good amount of ATP to make more amount of genetic material. Why? Because they are duplicating. That is why they consume their glycogen material and they send it to lactic acid cycle to produce ATP. Okay, so that is glycogen rich. So yeah, if there is abnormal precancerous areas, there is no staining of the Lugol's iodine, but rather yellowish white patches will be seen. So that was all about the pap smear, VIA, visual inspection with acetic acid, and visual inspection with Lugol's iodine.